Okay, first I would like to thank Dr. Nagla for the invitation to visit this very nice country and to meet all these very nice people that I've met since several days. I'm not a GM crop person. I'm coming from France. So I added a bit on my title, and I think the most important things I'm going to talk about is what is written in red, talking about insertion mutagenesis and how we develop this in Medicago would take me maybe 2 hours. So I'm going to talk about a plant, which I think in the dictionary it said something like it's the most misspelled name in biology, and we just had an example. This is Medicago truncatula and not truncatula or whatever. This was just a joke. What is Medicago truncatula? It's a relative to Medicago sativa, and this is the main interest. But because you know Medicago sativa is tetraploid, it's difficult to do genetic with this plant. In my lab, we are studying plant-microbe interaction and symbiosis because I believe symbiosis is important and plant-microbe interaction is important. We are still far away, despite what some people says, as far as I can imagine, to transfer this nitrogen fixation capacity to other plants. This is a fascinating area because in a nodule, a plant cell can host thousands of bacteria. In one plant cell, you can have over 1,000 bacteria. And this is the only example in the plant system that this can happen without eliciting a big defense mechanism. So it extends over the nitrogen fixation, and it's going to be important in next year to understand also plant microbe and pathogen interaction. I'm not going to go more on this. Medicago truncatula is all around the Mediterranean basin. It's not much used for agriculture, but it's a model plant. It has all the facilities for what we do in model plants. It has a small genome sequence, and we have plenty of genetic tools, so we can use it to discover genes. And I think this is one of our interests of this plant, is to bring new genes, new functions, and not understand only the plant-microbe interaction, but give new genes for the future. So when we started with this plant several years ago, or a long time ago, there was nothing known, and we had to establish transformation regeneration system. And this is, I spent a bit of time on that because if you want to do GM crops, or if you were to change crops on plants that are endogenous to your country, you have to be able to do transformation. And there's many ways to do transformation. And the way we do it in this legume plant is mostly through somatic abiogenesis. But I want to bring some points. Is there? Okay. Is there a pointer somewhere? Oh no, now I have the mouse. It's okay. No, don't worry. I didn't need this. What I want to say is that this was developed in different ecotypes, so it's going away. Nice. This is what is the pointer. On the bottom? Okay, thank you. Good. So this plant was not transformed before. Well, the regeneration capacity is based on recurrent selection in the lines we have now that are able to regenerate. Recurrent selections means then the people were going several times to tissue culture with the same lines, so that you improve it's kind of genetic selection for the ability to do, in this case, embryogenesis. This means that what you have at the end, you have highly regenerable lines, but this means, and this is what I tell always to the people, these lines we provide to the community, these are mutant lines, because they are different from the wild type at the beginning. And in our case, we use somatic embryogenesis. Some other protocols have been developed to do other type of regeneration, but this is, to my knowledge, the best, the most efficient in Medicago. And then when it goes to transformation, then you have to find a good agrobacterium. This seems to be obvious for the people who deal with transformation every day, but for those who start new system, they have to know that in this case you have to find the right agrobacterium strain. In Medicago truncatula, for example, we have one strain that we cannot use at all. This is the one that everybody is using for tobacco transformation, LBA 44. And then you have to find a good selectable marker. In our case, we are working with 2 lines, and not the 2 lines which are coming from 2 subspecies, but they are both Medicago truncatula. One is working very well with kanamycin, the other one is not working well with kanamycin. We have to use other marker, other selectable marker. Shortly, what is somatic embryogenesis? We start with leaf explants, we induce calli, and then from this calli we induce embryos, and we bring this embryo back to the to plant state, to the greenhouse. So if you have this, you can do transformation, and if you can do transformation efficiently, you can do mutagenesis. And because we wanted to do genetic in this plant, this was important to try to do insertion mutagenesis. Well, there's different things interesting in insertion mutagenesis, but the point is that what you want to do is to have tags that you can use to fish the gene of interest, and they can be either transposable element, they can be retrotransposon, or they can be tDNA. Everybody's using tDNA normally. And when I presented the beginning of this work, long time ago, people were telling to me, don't use transposon, don't use other things than tDNA. tDNA is working in Arabidopsis. Why should it not work in other plants? It's not efficient in other plants. And this is another take-home message. Every plant, you have to adapt a new system to it. And you have to be careful if you go because with your favorite plant that is used in your country, you have to try different things. If one system is not working, it may be normal. You have to go to another one. Every plant is different, so you have to adapt every time. We try to do the tDNA mutagenesis. You can make transformant, but what's happening is that if you get mutant out of it, none of them is tagged, or nearly none of them is tagged. So we are dealing with somaclonal variation. And then that's an old story. What is somaclonal variation? There were at this time many explanations for this, but we didn't know what they are. Despite this, there were some interesting mutants, and we wanted to follow them. So I will make just a short break in the following of this to say that insertion mutagenesis, we have success to do it in Medicago truncatula very efficiently, using not the tDNA, but using a retro element that's coming from tobacco. So we make, in this case, GM crops, because we have used something from tobacco to put it in Medicago. And now this retrotransposon is working very efficiently. The transposition is induced during tissue culture. We have 25 copies per plant. It's stable in the regenerated plant. It goes through the progeny. You can use as a genetic marker, and you can reinduce by tissue culture, which means you can expand the collection to a very large number. So this is a very long story that I put in 2 slides, and we collaborated with the Nobel Foundation in the state, and now we have regenerated, or they have, in the case of the Nobel, 20,000 lines. I told you 25 inserts per line. This is 500,000 inserts in the Medicago genome, and there's only 30,000 genes. So we are saturating completely the genome of Medicago. And there's a website there, you can get all the information you want on this, or I can give it to you later. So insertion mutagenesis is working very well in Medicago now. It's used by the community to discover a lot of genes. But the point that I want to make today, I used up half of my time really, is that, as I showed you just before, if you use tDNA or normal regeneration, and in fact we don't need the tDNA for this, we see that there are mutations in the progeny of these plants. We are going through somatic embryogenesis, and then we have mutant in the progeny. And because we were interested in this mutant, we went on with this story. So this is this mutant, bring back to nitrogen fixation. This mutant is not able to interact with the rhizobium. It's not seeing the rhizobium at all. So it was interesting for us to know which gene is involved in this. There was a tDNA in this line, but it was not linked to the mutation. So the mutation was independent of the presence of the tDNA. So then we had to do what we didn't want to do when doing insertion mutagenesis. This means map-based cloning. We map-based we positioned the mutation on the genome, and it turned out rapidly that there was in this region a gene that we knew already that was important. And then they say, okay, now we know what will happen with this. And the student, it's not a big gene, the student PCR, the complete gene, in the wild type background or in the mutant background. These are 3 individuals coming from the same mutant plant, and what you see is that we have a big shift in the size of the gene. It means there is a big insertion in the gene. Because we work with retroelement, this was an exciting day when she got this result. That we say, okay, maybe we have something interesting. We sequence it, and what is inserted in the gene is a retroelement. Again, like the one we use from tobacco, but this one is a Medicago truncatula retroelement. So, and it's a copia type element, and there's, this will come later in a few copies. So it's not very big. And it's inserted in this gene after regeneration from the Medicago plant. So just a short description. These are plant copia type element. And you see that they are all grouping in the same region. What is interesting is that, probably we cannot read on this, but the one, we call it MER for Medicago retroelement. And this is in the same group as the TNT one from tobacco. And just what I said to you, this is 1,000 blot from different plants, and this is PCR-based detection, so we are amplifying the border of the element. What it shows is that for retroelements, this is a low copy number. Normally you have thousands per genome. This one is quite low number. This is a wild-type plant. This is another wild-type plant from another subspecies, and this is the mutant corresponding to this wild-type plant. And what you see is that the difference in copy number is not very high. Here there's a difference that probably corresponds to the mutation. Okay. So, but what is important is that the mutant is slightly different from the wild type. So this is the way we unravel this. Just as a reminder, I'm talking about a retroelement, and the difference between the retrotransposon and transposable elements, as described by Bamar and McClintock, for example, that so the DNA-based class 2 transposable element. They transpose through a mechanism that's called cut and paste. They can excise from the locus and they reinsert somewhere else. If they excise, they eventually leave a footprint, and then they go somewhere else. So you start from one element, and after transposition, you have one element. For the retrotransposon, this is different. You make there's no excision. You make an RNA copy. You do reverse transcription, and this is happening in the cytoplasm, the reverse transcription. and then it goes back to the nucleus, it inserts in the nucleus. But because there's a cytoplasmic phase, it doesn't know where it was at the beginning, and it's spread over the genome. But the main result is that you start from one and you have many. And that's 1,000 blood to show that the transposition of this element is probably linked to tissue culture. And this is so the message for today. If you regenerate plants, so each line corresponds to individually regenerated plant. And this was with one transformation, this is another vector in this case. And what you see everywhere, you have a star. So this one looks like the wild type, this one is a new insert, this one is a new insert. They are probably siblings in this case because they have the same one. This has more inserts, this has more inserts, this has more inserts, and so on. And you have some that have quite a lot. This one is a new one here, this one is a new one here. Actually it was 2 inserts. So this is random effect, it's insert, it transposes not all the plants, but half of the regenerated plants, they have new inserts. So when you transform, make a transgenic, you have one chance of every 2 plants that you have activated transposition. What I showed you before, I think it's in, well here, belong to a small gene, no, it's not gene, but small family of transposable elements. 4 of them have a full capacity to code and transpose. We made specific oligos, but only one of them in the genome is transposing. And what is fascinating is that we have 2 subspecies, and the same copy is transposing in the 2 subspecies. Okay, why does it transpose during tissue culture? So the question is, when is the next question is, when is this element expressed? And this is where this is 1,000 blot in the 2 subspecies. This is 2 days of tissue culture. This is 1 week of tissue culture, 2 weeks and 28 days. 2 days of tissue culture, 1 week, 2 weeks and 28 days. And you see that in both cases you have a transient expression and then it goes down again. So it's activated early during tissue culture. And this activation, we went a bit more in detail, is linked to demethylation of the element. So during our process of tissue culture, what's happening is that when we de-differentiate the cells to get the calli to be able to do somatic embryogenesis, what we see is that the CG base pair, after at day 7 and day 14, they are less methylated than at the beginning of the experiment or after 4 days. That just happened to correlate perfectly to the time of expression of the element. So it's manipulating for regeneration. In fact, what we are doing is that we are manipulating probably the whole genome through silencing modification of silencing state, and that in this case we activate this element. How much time do I have left? It's okay. 2-3 minutes. That's good. Then what we have done, when we do this, these are PCR-based technology. So these fragments, they correspond to borders. I didn't explain it in detail. These are borders of the element. So it reflects the copy numbers of the transposable element in the genome, and these are the new borders after transposition. But because these are PCR-based fragments, it's easier to reclone and to know where the gene is expressing, where the transposable element is inserted in the genome. So by this way, we have recloned many borders from the element to have an overview of where is the transposable element inserted. Oh, sorry. And we have done, we have also, and this is also publish all the information for the TNT1. And what you see is that for both elements, so the endogenous retro element or the exogenous one that we put in Medicago, the inserts are around 28 to 33% encoding sequence. And what you should know is that this is the double of what is expected. So these elements, they have a not only they transpose in the genome, but they have a tendency to go in genes, and it's double as much as what we expect. Okay. So the conclusion on that, and that will be the take-home message for you about transgenesis and making transgenic plants through, at least through this method of regeneration. This transposable is a copia type retro element from Medicago. A transposition is linked to methylation of the DNA, a process demethylation that's probably induced by tissue culture. Its low copy number in Medicago truncatula. It transposed during in vitro culture. What I didn't show you here is that it's transposing only during tissue culture and only during these 2 weeks. That's it. We never saw transposition outside tissue culture. And it inserts in gene. So for us doing insertion mutagenesis, this is a very nice tool that we can add to the other one, but it's unrelated to here. But what is more related to this conference is that, as far as we can tell, the activation of this retro element is responsible for the somaclonal variation of regenerated Medicago truncatula. We have other plants that have mutation that we found, and for the moment, I don't say it's always the case, for the moment this is linked to this element. So most of the somaclonal variation in the regenerated plant can be explained by what we see here. And for those who are interested in the detail, this is the publication. I put it here. Reda, who is talking after me, was involved in also in this work.